Good morning, everyone. Welcome to the September webinar for the Atlas of Living Australia. My name is Martin Westgate. I run the Science and Decision Support team at the ALA, and I'm really thrilled to be here. We've got a great webinar for you today, but before I launch into that, I'd like to acknowledge, while normally I'd be calling from the lands of the Ngunnawal people, I'm interstate today, so I'm calling from Sydney, from the lands of the Gadigal people of the Eora Nation, and I'd like to pay my respects to elders past and present. Yeah, just really thrilled to be here today with three speakers from all around the country. So look, the topic of today's webinar you'll have seen from joining in today, whether you're logging in from in the future on webinars or whether you're watching live. And it's about technology. Now, that may not be entirely surprising. We've been doing these webinars for coming up on 4 years now. A lot of interesting technology talks, but something that's particularly exciting to me about today is that it impinges on an area that's very close to my heart, which is about ecological monitoring. Look, to say that technological development is moving quickly is sort of an obvious statement these days. And yet in ecology, the ability to detect species in the environment is increasing rapidly and markedly. And so I'm really thrilled to be able to hear from three experts today in that field and to talk about different technologies that all have that shared goal. And I think are really genuinely revolutionizing the field and environmental monitoring in Australia and around the world. So we've got three speakers today. We've got Christine from Macquarie University. Give us a wave, Christine. We've got Paul calling in from QUT. And we've got Matthew calling in from Wild Obs at the University of Queensland. Now, for those who are watching live, we have a questions portal. You'll be able to see it on your screen. We also have a poll at the end if you want to tell us something about yourselves. We'd love to hear about it. It always helps us to customize our future webinars to you and your needs. But with that in mind, I'll get out of the way. Our first speaker is Christine, and so I'll hand over to you.
Thank you so much, Martin.
No worries.
Just get this.
All right, that's looking great.
Perfect. So good morning, everyone. As Martin said, my name's Christine Chivers. I'm a PhD candidate from Macquarie University. And today I'm just going to be giving you an insight into some of the work that we've been doing on monitoring birds and mammals in Kakadu using minimally invasive approaches. I'd first like to begin with an acknowledgement of country. So today I'm tuning in from the lands of the Wadawurrung clan of the Darug Nation. I'd also like to acknowledge the Mirarr people whose lands of Kakadu I am fortunate enough to be able to do research upon and extend that respect to any Aboriginal or Torres Strait Islander people present today. So first and foremost, why do we need novel and new approaches to biodiversity monitoring? So currently we're seeing a global biodiversity decline, and this has been particularly pronounced within Australia's loss of mammals. A third of all modern mammal extinction events have occurred in Australia, despite only being home to 6% of the world's mammal diversity. This has resulted in 39 mammals going extinct since European settlement and listed a further 110 as at risk of extinction. Despite Australian birds faring better, we do have 163 birds currently listed as at risk of extinction and have seen a 60% decline in abundance of these certain birds over the last 40 years. This brings me to next-gen biodiversity techniques. So ultimately, the first step in being able to temper this rate of biodiversity decline is being able to gain reliable and robust biodiversity monitoring data. So today I'm going to be talking about three of these techniques: passive acoustic monitors, camera traps, and eDNA or iDNA-based approaches. So people are probably more so familiar with the first two on that last slide, but not so much familiar with the eDNA or iDNA side of things. Some people may have heard of the term eDNA, which stands for environmental DNA. Well, essentially iDNA is a subdiscipline in this field and stands for invertebrate-derived or ingested DNA. It relies on the capture of invertebrate samplers, so things such as carrion flies, leeches, and mosquitoes, but essentially through the day-to-day feeding are collecting that vertebrate DNA for us. This offers us the opportunity to collect these invertebrates as a means of passively sampling the vertebrate diversity of an area. So far, the approach has shown great promise in its ability to detect multiple vertebrate groups with one method, an approach that can be challenging with traditional techniques, as well as increased sensitivity and difficult to monitor groups, including small-bodied and arboreal mammals. And studies also demonstrated its ability to detect more mammals than established minimally invasive techniques such as camera traps, as well as in some cases being able to differentiate between cryptic or morphologically similar species. So this brings me to what we've been doing in Kakadu. So we have sampled 6 1-hectare reference plots on the northeastern edge of Kakadu National Park during 2 rounds of sampling undertaken during a wet and dry season. We have employed these 3 milli invasive techniques, so acoustic monitors were only deployed during the dry season, camera traps for a 6 to 8 week deployment during each field campaign, and mosquito eDNA for 2 consecutive nights of sampling at the beginning of each of these field campaigns. So this is just an idea of what our setup looks like. So we have an acoustic monitor in the middle programmed to record for 2 hours in the morning, 2 hours in the afternoon to capture that dawn and dusk chorus, camera trap baited with some rolled oats, honey and peanut butter, and a CO2 baited mosquito trap that essentially pulls the mosquitoes in and they're sucked into that net that we come along and collect the next day. So as you can see, we definitely had no shortage of mosquitoes in Kakadu. We were getting on average between 800 and 1,500 mosquitoes per trap and collected over 48,000 mosquitoes across the 2 field campaigns. So what do you do with 48,000 mosquitoes? Well, essentially iDNA follows the same sort of meta-barcoding workflow you would in an eDNA-based study. To simplify, it starts with a DNA extraction process, in our case from bulk mosquito samples, amplification of this DNA using mammal, vertebrate, and avian primers that then goes off for next generation sequencing. In return, you receive millions of reads back that essentially go through a bioinformatic and taxonomic assignment process. So what do these techniques tell us about the detection of birds? So just to clarify what we mean by sampling effort on these species recovery curves. So acoustic monitors is the number of trapping periods per trap. Due to time constraints, we were only able to analyze the period that corresponded with the collection of iDNA. iDNA corresponds to each of those bulk mosquito samples, and camera traps is the number of days of deployment per camera trap. So as you can see from this, we're getting significantly more bird detections with both acoustic monitors and iDNA than with camera traps. So in total, during the dry season, when we had all 3 methods deployed, we were able to detect a total of 79 avian taxa. This included 52 with acoustic monitors, 49 with mosquito iDNA, and 11 with camera traps. Only 6 of these were detected with both methods, while 21 were detected by both acoustic monitors and camera traps. Despite this, though, there was still a considerable amount that were unique to each method, essentially telling us that these 2 methods are additive and will be able to result in the detection of additional taxa than they would be on their own. Just to put into summary some of the examples of some of the birds that we were getting with different methods. So with the acoustic monitors, we found we were detecting additional honey eaters, as well as transient species, so things such as the magpie goose and red-tailed black cockatoo, that may be passing over our sites and getting picked up by these monitors, but aren't necessarily stopping to get fed on by mosquitoes. While the taxa that were detected with both methods included relatively common birds, so things such as the blue-winged kookaburra, tawny frogmouth, and galah. While with iDNA, we found we were getting more ground-dwelling birds, so things such as the brown quail and the threatened white-throated grass wren, as well as semi-aquatic birds, so things such as the Nankeen night heron and intermediate egret. Generally, aquatic birds can be challenging to monitor using approaches such as acoustic monitors. So what about mammals? So we found we were getting pretty much double the amount of mammal detections with iDNA as with camera trapping, with pretty much almost a quarter of the sampling efforts. So with camera traps, we were able to detect a total of 9 taxa across both field campaigns, and these included relatively common ground-dwelling species, so things such as dingoes, agile wallabies, feral cats, and the northern brown bandicoot. Despite this, though, the prolonged deployment of the camera traps over that 6 to 8 week period did result in the detection of additional species that weren't picked up by iDNA, most notably the threatened black-footed tree rat. While with iDNA, we were detecting a total of 20 taxa. This included additional boreal species that aren't picked up by camera traps, so things such as the sugar glider and ghost bat. Small mammals, so things such as the pale field rat and common rock rat, and as well as additional macropods, most notably the short-eared rock wallaby at one of our sites that is near the sandstone escarpment, and the relative elusive spectacled hair wallaby. So what about reptiles, you might ask? So we found in this study that although mosquito iDNA appears to be a good approach for mammals and birds, not so much for reptiles in our case. We did find that camera traps was able to detect the most reptiles. However, these were mostly large and diurnal species. So you can see down on that left-hand corner, we've got a frilled-necked lizard, and in the right-hand side of that right-hand picture, we have a yellow-spotted monitor. And currently, studies do demonstrate that currently a combination of camera traps to capture those larger species and live trapping for smaller and nocturnal species appears to be the best approach, really hiding that there's still work to do and being able to develop many invasive sensitive techniques for the detection of reptiles. So as with any approach, all of these methods do have their own sets of challenges. So a correct identification can be challenging with all of these methods. So acoustic monitors, you can have challenges with incomplete calls, crossover, or species variation in calls. Camera traps, you can have issues with blurred imagery, especially for small, fast-moving species and morphologically similar species. So to put this in perspective, with iDNA, we were able to detect 3 different rat species. This included the introduced black rat, as well as the common pale field rat and common rock rat. However, we were challenged to be able to get any species level identifications for any of our rat images from camera traps. iDNA can also have its challenges. So essentially any iDNA or eDNA based study is really only as good as the reference library that you have to be able to match those sequences to. Furthermore, all these challenges do have their own specialized skill sets. This range from the molecular skills required for iDNA-based approaches to the localized knowledge for the identification of acoustic calls. However, in saying that though, both the use of AI as well as the commercial labs offering eDNA services are making these approaches more accessible for a wider audience. Additionally to this, we do have challenges with the transportation of CO2 to be able to capture mosquitoes, particularly when working in places such as Kakadu. And you do always have challenges with equipment failure and destruction. So over half of our bait stations were destroyed during our campaigns. I'll give you best guesses who may have been responsible. We've got suspect number A down in that right-hand corner. So just to summarize this all, so we found that mosquito DNA outperformed camera traps in the detection of mammals, detecting additional macropods and arboreal species. We found that acoustic monitors and iDNA appear to be complementary approaches, with acoustic monitors resulting in the detection of more honey eaters and transient species, and iDNA more ground-dwelling birds and semi-aquatic birds. However, camera traps appeared to be at best for reptiles in this study. However, our current approach of live trapping and camera trap appears to be the best approach, with that really demonstrated need to continually to work on new sensitive techniques for reptiles. And furthermore, also the continued need to improve DNA reference libraries to really uptake that use of eDNA and iDNA-based approaches. So just to wrap up today, I'd like to acknowledge a few people. So obviously this research couldn't have been possible without especially my supervisors, Anthony Cheriton, Adam Stowe, and Andrew Harford. The support from Macquarie University and my lab and the eDNA and Biomonitoring lab, as well as our collaborators and project partners of the supervising scientists who this project wouldn't have been able to happen without. And special thanks to Tom Mooney, Dave and Lowensteiner, and Kate Montague.
That's wonderful. Thanks, Christine. Look, I've already got so many questions, and there's already some coming through the chat as well. Now, normally in these events, we hold questions at the end, though, so I'll queue those up. It's a fascinating talk, and thanks so much for sharing your research with us. Now, Matthew, over to you.
Thank you. Can you hear me?
Yes.
Great.
And the slides are looking great, too.
Thank you. So I'm very happy to present the Wildlife Observatory of Australia, which has recently been funded through ARDC, QSIF, TURN, and the University of Queensland. It's a big initiative with a lot of people that I will acknowledge at the end. So I also want to start by acknowledging the traditional owners of the land that I work here at UQ, and I'm going to hop right in. So what is WildObs and why do we need it? We are going to create a national wildlife database that will deliver information on robust monitoring, science, and management. We need this information on wildlife to track threatened species. We need it to track invasive species and management. We need it to produce high quality scientific innovations. We need it for our public to be able to access information about where their amazing wildlife is. And we need it to meet our UN biodiversity goals that we have committed to as a country. We also need it because there are dramatic events and these require timely information so that we can manage them. These include things like bushfires, disease outbreaks, climate change, and we want to be able to sell biodiversity credits alongside our carbon credits, which means we have to be able to robustly, quantitatively track wildlife. So we have some opportunities and some problems with cameras. So cameras are relatively easy to set, strap them to a tree, leave them for a few months. Easy to set 50 cameras per survey, but these create so much data, hundreds of thousands of images, over 100 gigabytes per survey. And we have many surveys in Australia, but these are completely uncoordinated, often targeting a single species, but they capture so many species that if they were brought together, we could use it as a monitoring tool. We are collecting a lot of data, but we are not publishing as much as we would expect based on this investment in our sampling. This basically boils down to camera traps are easy to use in the field, but generate unmanageably large data streams, and these data streams are not being leveraged to produce high quality science and monitoring. So who is using cameras? Well, basically everybody. They're very accessible to researchers at all stages in their careers. They're used in a wide variety of biodiversity research, management, and monitoring. People that use cameras in Australia include NGOs, government, consultancies, academics, and indigenous groups. So right now, camera data is being boiled down to presence only. This is not the richest format for the data, but it is suitable for species distribution models, something that Australia really excels in. But species distribution models do have some limitations. They're really good range maps, essentially, but they just tell us if the species is there, how likely it is to be there. Well, camera surveys actually include the detections and non-detections. This enables a type of modeling called hierarchical modeling to derive occupancy and abundance while accounting for variable detection. What that means is two people can set cameras, one with baits and one without, and we can account for the fact that species are detected at different rates utilizing different methods. That's extremely powerful for robust monitoring. It allows us also to look at trends through time and to infer species interactions, both of which I will show you examples of. We also want to note that while ALA focuses on presence-only data to date, we are in process of making it more accessible to download full survey data using the same website that we all know and trust to go to for our biodiversity information. So right now, only a third of Australian publications utilize analyses that account for detectability. This is relatively low, and it could explain why our comparatively low publication rates compared to other places. So what are the things WildOb is going to address? We're going to enable coordination and collaboration in sampling fieldwork so that we can be more productive and efficient and save money and answer our questions more efficiently. We are also going to help with image management. So this is the storage and efficient processing of images. This needs to be free, needs to be cloud-based so we can access images anywhere we want. We can have many people sorting images, and we need to be able to power AI identifications, and I'll explain that in more detail. We also need open access to data sets. Right now, these are often siloed away at specific institutions or in someone's old dusty hard drive. This is especially important for publicly funded efforts. So anything from ARC, federal government like DQ, state governments, these are all required to be open access, but they currently are rarely open access. We also need training in advanced statistics for impactful research and monitoring, and this is something that we are going to be really focused on. So in order to store, organize, and identify millions of images, we need access to a cloud-based image platform that has identifications using AI. One example is Wildlife Insights, but there are many, and we are debating which one right now that we're going to promote and provide free for all Australians. We also need tagged images in a repository so that our AI researchers here in Australia have what they need to do new AI research and improve our algorithms. We also need an aggregation of all the spreadsheet data that we're currently deriving in all these separate places, separate institutions, separate labs. So we need standards for our data, and we need APIs to share that data. And then we need it to be accessible and searchable, such as via online on ALA. Then we need robust analytics to drive key inferences. This requires meticulous data standardization and preparation, robust statistics, and we need to create intuitive results so that people can really understand what is being derived from their sampling. We want to create automated reports. You provide us data, we give you back an automated report that includes the best robust analyses possible. We're going to feed those analyses and the results into the government regulation like the EPBC Act for endangered species and our indices like the Threatened Species Index, which I'll talk about in a second. We're also providing training for statistical skills. And we're initiating many more collaborative papers and reports. And this really is a carrot to get people to be involved because they can get a lot of publications if they're part of this group. As a proof of concept, we worked in Asia in our lab to put together 239 camera trapping studies, published a data paper. Then that paper has been used in over 20 publications within the last 2 years, including a publication that made the cover of Nature. So this indicates that if we can do this, there's really powerful science that can be derived. We really want to contribute to the Threatened Species Index. This shows trends through time for birds, mammals, plants. Right now, there's very little camera trap data in this because of a variety of reasons, and we can solve that so we can make that orange line extremely confident in what it's reflecting, and we can do an orange line for every single mammal species. I want to give you one example, which is the Eyes on Recovery project. This was thousands of cameras set after the 2019-2020 Black Summer fires. Massive amounts of data. We were able to analyze the images using wildlife insights, and the AI helped make that efficient. And then we were able to process all that data in 8 weeks, provide specific, robust with hierarchical models for 93 species. And today we can do that in 1 to 2 weeks. This is what we need to provide timely, management relevant information. We can also use these richer data sets to look at species interactions. Here's an example from Tom Bruce, who I'm going to give you a little more information about his work in our lab. And what it shows is the green line is the dingo activity throughout a day as captured by camera traps. That blue line shows how cats behave in the absence of dingoes. That red line shows what happens how cats behave when dingoes are present, showing a strong shift toward nocturnality. So these sort of inferences cannot be gleaned from presence-only data. We're expanding that type of research to look at entire food webs. If you want to learn more about this, Tom's actually giving a seminar next week. We're going to drop the information for that in the chat. And Tom's work in the lab, managing all this data, has a few easy conclusions. That setting cameras going out in the field is really fun. Managing 25 million photos is not fun, but we are there to help. So some of our progress to date is that we've already had a review paper with 194 Australian authors representing all major universities, federal and state governments, and NGOs. We've also been collecting data, I should say collating existing data. We're already up near 30 million images. We have a very defined data standardization process. This is led by Dr. Zach Amir, and we are nearing a choice of platform to provide for all Australians that will provide free, limitless image storage, easy management, and AI identifications exceeding 95%. We're targeting 98% so that we don't even have to review them. Once we get to that accuracy, we don't even have to look at our images. We just trust the computer is doing a great job. We're also working with DQ to provide a metadata collection app. This is going to be called Monitor. You'll hear more about it in the next year. This is going to allow everyone to enter data in the exact same format from their smartphone in the field, and that will automatically upload it to the cloud. The government can see where you've sampled. It provides everything you need for reporting, and that is linked to the image outcomes later. And finally, we're hosting statistics seminars, and we're providing statistical tools. So if you have data, need help analyzing it, we can help you. I'm going to let you under the hood just for a second. So first, I'm going to explain our pipeline for images. So images are sent to us either as SD cards or uploaded in the cloud in a Dropbox. They are moved into what we call a WIMP, a Wildlife Image Management Platform. Examples of this include Wildlife Insights, and there's even one being developed here in Tasmania by Barry Brook called Muke. So this platform hosts all your images, allows you to see which cameras collected each images, allows you to sort per for different species. And what it also does is it puts all identified and unidentified images through something called megadetector. That tells you what has an wildlife versus anything else. If it has wildlife, it goes to a computer vision classifier, which is an AI tool to identify species. If that species is identified over 97% accuracy, it goes straight as data. If it's less than that, it gets verified by a human. All that data then goes into our data standardization process and our camera database. Okay, now I'm going to shift this up to show you some more. If you already have data, or your institution already has data that's been sorted, or you have your own image management platform, we can pull that directly either from your platform or through ALA. They have an API, and that goes also into our data standardization international database. We also are pulling directly from biodiversity aggregators, so Queensland, New South Wales, BioNet, WildNet, federal government, BDR, Biodiversity Data Repository. This is where a lot of data already exists, but it's currently siloed in these places. And right now we cannot pull full detection histories, only presence only, and so we're changing all that. So we are providing external access through ALA, and this is what is going to enable wildlife biologists and government officials to monitor biodiversity easily. We're also allowing a tagged image repository where we keep all these images collated in a nice format so that can be used directly by AI researchers. So we can be on the forefront of developing these tools. So with that, I'd like to thank many people listed on this slide and thank the funders and the website to learn more about work that we already have done. For example, in Asia is at our website, on my website, ecologicalcascades.com. And if you want to learn more about WildObs, the website is turnslot.au.org/wildobs. Thank you.
Thanks, Matthew. Fascinating to see, and nice to see that other people have problems with too much data at times as well. It's something that hits us a lot at the ALA. Paul, you're our last speaker today. Would you mind sharing your screen? That looks great. Thank you.
Excellent. Okay, thanks, Martin. Thanks to the ALA for inviting me to give a talk. And it's great to talk after Christine and Matt, because a lot of what they've already talked about in terms of sort of the motivations and things are exactly why I'm excited about ecoacoustics. So my name's Paul Rowe. I'm an academic at Queensland University of Technology, and for the past 10 years I've been on really this journey into ecoacoustics. And what I want to do today briefly is to talk about a couple of projects I'm involved with. I would like to start off by acknowledging the traditional owners of the land where I now am, or where QUT is. That's the Turrbal and Yagara as the First Nations owners, the traditional custodians of the land which was never ceded. I'd like to pay my respect to the elders, laws, customs and creation spirits, and recognise that these have always been places of teaching, research and science, and the Aboriginal and Torres Strait Islander people play an incredible role in all of our communities. Okay, so I'd like to start by just talking a little bit about ecoacoustics and what it is. So really, ecoacoustics is a new way to understand our world through continuous sound recording. It provides a sort of a scalable way of monitoring biodiversity through continuous sound recording, and provides a direct environmental record of that. So it's a little bit like sort of remote sensing, drones and satellite imagery, which collect sort of images of of the continent, of the vegetation, and from those images we're able to deduce what's happening to the sort of forests. We can look at kind of flowering event and understand the sort of vegetation. But really, up until now, the problem has been that we haven't had scalable methods for understanding faunal biodiversity, and that's why we're so interested in things like camera traps, the ecoacoustics, and the eDNA. Ecoacoustics is a rather young discipline. There are a number of unique issues. Matt sort of alluded to some of those. Some of the issues are the same that we face in ecoacoustics as with camera traps. We've got sort of a lot of data. It's collected all over Australia, and we need to sort of bring it together. And also, it's quite difficult to analyse. There's been some quite rapid progress over the last 10 years, and I want to talk about a couple of those projects I've been involved with. And Australia's been taking a leading role. Before I do that, though, let me just play you some sort of sound. It's sort of somewhere collected on a property near Brisbane, and this was sort of a beautiful morning chorus there. So one of the great things about working with sound is you get to listen to these beautiful choruses to the beautiful birds, not just birds, but also frogs and some mammals vocalizing like that. Of course, the issue is how do we make sense of that? So one of the projects that I've sort of created is called the Australian Acoustic Observatory. This was a collaboration with some colleagues from other universities around the country. And what we set out to do was really to realise that vision of the kind of continental scale biodiversity monitoring, to really understand how can we monitor faunal biodiversity at scale? How can we actually understand what's happening? So we set out and created a project. We were successful with getting some funding through the Australian Research Council, and we set up a large sort of network of recorders. And one of the things about ecoacoustics is we regard it as being very much a data-first approach to science. So in that sense, it's a little bit like astronomy or bioinformatics, where you might actually collect data and you might start with that data and mine that data to actually sort of come out with, to understand what's happening, to find questions. So it's a data-first approach, and hence the name Observatory, because we're observing or listening, if you like, to all of the sounds across Australia. We are also very driven by an open science methodology, in that we believe that the data should be, it's been funded through an Australian government research grant. We believe that the data should be made sort of freely available, just like the observations through the ALA are. And we're using the data to support, obviously, sort of ecology and computer science and citizen science, but there are also creative uses of the data. As you can imagine, with such beautiful recordings, there's a lot of sort of artists and other people that are interested in using that data. We started deploying recorders in 2019, which is, of course, a great time to start. So we had a few delays due to bushfires and COVID and things. But we now have over 365 sensors deployed all around Australia. We're continuously recording sound, so there's no sort of sampling involved. This is continuous sort of sound recording. Each of the recorders is a little sort of solar-powered box that records to a set of SD cards. And we're very much leveraging the goodwill of landowners to collect the data for us, to collect the SD cards, dust off the solar panels, and send the SD cards to us. So we have this huge amount of data rolling in. The difficulty, of course, as Matt's already said, is how do we manage all of this data? We obviously want to get the data into the atlas, but how do we sort of get it there from, you know, remote areas like the sort of Sturt Desert, you know, bunch of sort of SD cards that are sent to us. We need to get that into some database where it can kind of be analyzed, and then the observations actually put into the atlas, so we can kind of generate those environmental insights. So to do this, we, along with some of my colleagues, created a project, an open source platform. This was supported through the Australian Research Data Commons, to essentially kind of complete that pipeline, that workflow for processing the data and to get the data into the ALA. It's been very important for us to work with sort of communities. So we are co-designing the tools and systems that we're producing. All of the tools and systems are sort of open source for everyone to use. So it's very much a program to a project to produce both a platform, but also to bring the community along with us, because the young discipline, so that we can understand how best to use the data, how people want to use the data, the tools that they need to deliver on this vision of being able to understand what's happening to our beautiful country. once we've got all this data, of course, there's still this issue of how to find calls. So, you know, somehow we needed to search through all the data. So we thought, who are we going to call when we need to do a search? So we've got a collaborative project with Google where we can actually undertake a kind of a search. So if you've got an example call that you want to find, we can actually scan through all of the data through hundreds of megabytes of data, hundreds of gigabytes rather of data to actually find those particular calls. And if you want to, perhaps take a photo with your phone or something, or just grab that image, or if your phone allows you, go to that link for the QR code, and you can actually give the similarity search a go. And this is all driven by Google AI. So just to sort of wrap up, couple of projects. We're collecting data with the Australian Acoustic Observatory from all over Australia. We've got other specific projects which sort of target threatened species or other monitoring regimes which various sort of NGOs and governments are running. We've got sort of literally thousands of users. We've got over 200 projects, over 0.5 petabyte of data now. Almost 500 years of continuous data, which obviously is too much to listen to, which is why we need the open Ecoacoustics platform to enable us to sort of store and analyze the data. And also with such big data, you get a lot of errors. We've got a lot of sort of very sophisticated tools to fix the errors. But as much as this is about data and about platforms and things, it's also about people. So I would like to thank all of my colleagues and collaborators that have made this happen. And I'd like to thank the ALA for inviting me to give this talk. So thank you.
Lovely. Thanks for that, Paul. I'm slightly unsure where to go. We now have about 20 minutes, which is a wonderful amount of time for a discussion. So thank you all for your presentations. People have been queuing up questions in the chat, and I'll try to get to what I can and summarize what I can't. So I guess to get started then, look, it seems like in each of those three talks, you've benefited from some pretty massive leaps in technologies in the last few years. Matt, you talked about processing things with machine learning and AI, and Paul, same. I guess the eDNA boom has been well documented as well, like the sort of technologies for genomics. Are we at the is everything else keeping up? You're now generating all this data. Do we need new development in our statistics to manage this kind of data, or have we really just been waiting for that? Sorry, just open question there if anyone wants to jump in.
I think this could be a good opportunity for me to jump in, because the statistics for wildlife ecology have largely been developed for cameras that came on the scene a little bit earlier than acoustics and eDNA, but can be used for those methods. For example, I mentioned these hierarchical occupancy and abundance models. Those take detection histories. So we need detection versus non-detection in each amount of time at each sensor. That can be created through multiple surveys of eDNA or through multiple surveys of acoustic data. The statistics are there. They're a bit complicated, but there's packages being developed in R and other tools that make them accessible. And one of the things that we're doing is producing automated reports that include all those complicated analyses that we run on our supercomputers here at UQ and QSIF, so that users who don't have a degree in statistics can still have access to robust outcomes from their data.
No, thanks, Matt. Paul, do you want to add to that at all?
Well, I'd probably defer to Matt. I'm not a statistician, but yeah, certainly. I mean, with the new developments in sort of hardware and software, so as you're saying, Martin, the sort of the AI developments, which is both the sort of compute and also the techniques to implement the AI, those deep learning networks, and the decreasing price of sort of storage, that's enabled us to collect and analyze more data. And as we get more data, we are going to, you know, potentially better find out and understand new things, but it is going to require, I think, new kinds of statistics, or certainly new kinds of models. So I think things like, the occupancy modeling and hierarchical models that Matt sort of talked to, I mean, those things perhaps have been around for a little while, but we haven't really been able to use those because we just haven't had the data. The great thing about, putting out, iDNA or camera traps or ecoacoustic recorders is that you're not reliant on a person being out there in the field making all of those observations. And so, yeah, it is going to enable a whole new kind of science, and that's obviously really important for conservation and understanding our environment, as well as, producing food and all of the other ecosystem services that we're reliant upon.
Yeah, no, it is as someone who did a PhD in a long-term monitoring lab where every observation was someone standing in the field. It's amazing to see the sort of wealth of data that's coming through. Christine, if I could pick you up on this question of dealing with data. I mean, it was a really stark example of a long-standing problem I felt in ecology that you demonstrated there, which is that different methods detect different things. How do you see this playing out in future? Do you think that we're going to have to, presumably there's a lot of testing needed to compare these methods?
Yeah, so I definitely think there's more work required to really understand the biases, especially associated with iDNA as sort of a neuro approach. So definitely understanding that and using that in a decision-making framework, and when planning biodiversity monitoring, working out sort of what focal groups you're wanting to focus on and how to go about that. Yeah, obviously there's still a lot more work, especially with iDNA, that needs to be done. There's a lot more questions than answers at the moment, but yeah, it's definitely showing great promise at the moment.
No, it's fascinating, and it's really good to see that work being done as well. There was a follow-up question. Oh, sorry,
Martin, I want to just bring up a frontier, which is that these hierarchical models, because they allow you to include a detection covariate for each survey, they also allow you to merge eDNA, acoustics, and cameras, with each of them having a different detection probability. So, for example, Christine mentioned how there was a vastly different detection of mammals, reptiles, and birds using these methods. Cameras excel at vertebrates on the ground, mammals predominantly, ground-dwelling birds. Acoustics excels in birds, particularly, and insects. And eDNA covers a lot of the most cryptic species that are hard to see on a camera, don't make a lot of noise. Putting them together in a single model, especially these multi-species, multi-site models, allows you to learn about an entire community in ways that we've never been able to do before. This is the frontier. There are models already available. It's not easy, but it's exciting, and it is the future.
And presumably the scale that you've got, the amount of data that's putting into these models, and the degree of standardization as well, is helping with that, I'd imagine.
Absolutely. What I think the most important innovation there is in this space is the ability to include species specific, detector specific, so whether or not your DNA, acoustics, or camera specific covariates for each of these sampling approaches, and then that allows you to bring them all together under a single modeling framework.
No, it's fascinating to see. It'll be good to see how it flows into things like the TSX, for example, that you mentioned, Matt. Look, I'm going to try and get through some of the questions that have come in from people who are online. And one of them, sorry to go back to you, Christine, so soon. There was one that was asking about co-benefits. So, for example, whether it's possible to detect pathogens from mosquitoes simultaneously with wildlife. Is that something that's on anyone's horizon?
So that is definitely possible. So you can do DNA and RNA co-extractions if you're wanting to look at that as well. So in our case, we were just focusing on the DNA, but that's definitely a probability that you can also do the RNA extraction to be able to isolate any mosquito-borne pathogens as well.
Wow. I dread to think the amount of data you'd get from something like that, but I imagine it would answer some very exciting questions. There was a similar one, Paul, for you about whether ecoacoustics is feasible in the marine environment, which I wondered if you had any comments on that.
Oh yes, there's a there's a sort of a long history of using acoustics for cetaceans and things. I was also going to jump back to the previous question, if I may, to say that there are other things that you can use acoustics for, because it does detect other information. So potentially you can get information about, you know, wind and rain and things, but also you can detect, you know, potentially, you know, if you've got some sort of human disturbance, you know, sort of gunshots and things like that, or, you know, motor vehicles are actually sort of noise pollution, which of course may itself be affecting the environment. So yeah, these methods, you can do a lot with them and detect more than just a sort of a single target species. You really can get a bit of a handle on what's happening to the whole environment.
And how far are we along on that journey? So there was a specific question in the chat. Someone was asking about mimics, so birds that copy other birds. Presumably, we're still at a point where some of that stuff is challenging for these models. Is that fair to say?
Yeah, that's a great question. We have done some work with mimics, particularly with Albert's lyrebirds, and generally, if you know, I think we're at the stage where if a human can perceive the difference, then usually, providing we've got enough, you know, test and training data, we can then actually train an AI model to distinguish the two. So certainly, we had success with the sort of Albert's lyrebird, but yeah. Obviously, there are limits to you know, what you can do, and sometimes you know, I think as Christine was saying, it's important to understand the bias of the technique. I mean, all techniques are biased, even if you've got a sort of a human observer in the field. Sometimes we can't distinguish particular species, so sometimes we can't distinguish, for example, between different raptors and things like that make the same kind of noises. Other times we may actually be able to distinguish individuals, so potentially with some birds and some koalas and things like that, we can actually get a handle perhaps even on the individual that's calling.
That's an interesting point, actually, because we've had a couple of questions in the chat about comparison against citizen science data, or I guess and with birds, of course, in particular. I'll pick on those just because we get a lot of data on birds and I see it a lot. Many of the people who are submitting those observations are extremely expert, you know, to the extent that they could be doing this professionally but don't. Do you have a sense of the extent to which automated methods, obviously they scale much better, they're in a sense cheaper per unit? Do you have a sense of their comparability against expert observers in the field?
I think they're pretty good. I mean, I think in a sense the advantage of a lot of these methods is that they can be deployed by non-experts, and then you can have experts review the data if you keep the data. I mean, even if you've got an expert birder in the field, you know, if you hear a weird sound, it's not a bird. You know, you hear a frog or something, and if you don't have a, you know, heptologist with you, what do you do? But if you've got the recording, you can then sort of take the data to the expert. So I think keeping the data is, you know, it's about the sort of scalability that the techniques provide, but it's also about the record, having the data which can then potentially be kind of audited and assessed and used for, you know, the sort of green accounting schemes. But also, if it's a call that perhaps not a lot of people know, or where you might really need an expert, you can take it to the expert. I see the AI as being an assistant, really. I don't think it's ever going to replace people. It's more an assistant to help people.
No, entirely fair point. Matt, if I could come back to you on this. So
I'll just real
yeah,
quick,
go ahead.
what Paul said is about the AI being an assistant. I agree with that. Traditionally, that's how we've used it. But for cameras, for a majority of species, but not every species, the AI is significantly better than a human, and the accuracy is much better than a human. So we're getting to a point where we never have to see the images, and we can have a workflow once we have, let's just say, universal access to a signal. The cameras can upload images, the AI will process them, and the data will flow into the spreadsheet so we can analyze it, completely removing the human from the link. And that will allow scalability on a whole nother scale. So I suspect that will happen for many species of birds in the future, maybe not all. I'm curious what Paul thinks on that one.
Well, I guess some bird calls evolve over time, so it's not a a sort of a static system that you can completely analyze. I think that the AI is going to greatly help with dealing with the massive amounts of data that we've got, but I think there's always going to be a role for a certain amount of validation and checking. I think we will also need to look at other techniques. The finding bird calls in a long recording is a very reductionist approach to understanding the sound. I think, and all you find out is what the recognizers have been trained to do. I think it's also useful to have other perhaps unsupervised approaches where you also try to kind of characterize the overall sort of soundscape. I mean, crudely, if a microphone gets eaten or damaged, as Christine sort of knows all too well with some of the problems that she's sort of had, the recognizers aren't going to tell you that. They're simply going to say that we haven't found anything. They're not going to tell you that, well, actually, there's no sound data there.
Yeah, I've all had that happen. I had a bushfire go through some of my sites when I was trying to record some things, and that was in the tape days, but let's not go back to that. While we're talking about this sort of scale, Christine, we haven't really talked about, you've mentioned eDNA and iDNA, and they're in some ways newer technologies, but also becoming quite established. Do you think they have the potential to scale to the same sort of thing as we're seeing in ecoacoustics and camera trap data?
Definitely, I think especially with the uptake and also like things like citizen science projects employing eDNA, that we definitely have the application to extend the use of it. So it's definitely getting more accessible and definitely increasing in its scalability, especially as well as I was saying earlier, with more commercial labs also offering eDNA services and that sort of workflow becoming more streamlined. Yeah, it's definitely increasing in that ability as well.
And you think with a body of users in the bioinformatics community who are used to large data already, sort of from straight off the bat, that would be a benefit.
Definitely also with supercomputing and like the increasing bioinformatic capabilities, yeah, it's definitely going to be easier to be able to process that data as well. There's definitely people that are working on ways to try and essentially take that data and have it come out in a format that's accessible by government agencies or people who might not be familiar, what a OTU table or anything like that looks like. So there's definitely things in the pipeline to try and make that as a data source that is more easily able to be understood by sort of general users as well.
Definitely some challenges there, though I'm sure some opportunities as well. Look, we're coming close to time, but I will try and sneak in one sneaky question, which and I'll direct it to you, Matt, first, if that's all right, which is that now, some time ago, a year or two perhaps, the Australian Academy of Science put out a report suggesting that Australia needs a Bureau of Biodiversity in the same way as we have a Bureau of Meteorology. Now, some of the things that the presenters today have put forward are what look like first steps towards something like that. Do you feel that in future there's the potential for an integrated monitoring system for biodiversity in Australia that could be productionized in that way?
Yeah, absolutely. That's in the cards. There's a variety of initiatives around biodiversity right now that are playing out. So the landscape's evolving really quickly. One thing that has been great for at least Paul and I is the Australian Data Research Commons, ARDC, has a new arm called ARDC Planet, devoted to large-scale data commons for biodiversity and environmental data. They've supported both me and Paul and are looking at expanding that to drones, potentially eDNA in the future. So the ball is rolling, but the path it will take is unclear at this stage.
That makes sense. And obviously we're speculating at this point, it's with the technology moving so fast, it's always hard to be sure. But it is interesting to think that these things would have been impossible a decade ago, for example. Lovely. Okay, well, with that in mind, thanks everyone for your time. I'll remind everyone who's watching along at home, we have a poll that we put out. If you want to tell us something about yourselves, that we're always glad to hear about it, please click on that on your screen if you'd like. But with that, thanks so much to all our speakers today and for those insights. This will be available on YouTube in due course. And enjoy the rest of your day. We'll see you at the next webinar. Thanks so much.